Welcome back to Lecture 2. This would be the second half on organic molecules. So recall previously that we've done inorganic molecules. These would be very small, typically non-carbon containing molecules. Organic molecules are different. Organic molecules are incredibly large, and organic molecules will always contain large amounts of carbon. Now the organic molecules are proteins, carbohydrates, lipids, and nucleic acids, and again, very large and will always contain carbon. So before we really get into the meat of the conversation today, we need to have a little bit of a conversation on synthesis and decomposition and the anabolic and catabolic reactions and just general building and breaking of things. Okay? And it's going to go something like this. Anabolism means to build something. You put energy in and you make something. Catabolism is to break something down, and this will liberate energy. It releases energy. Okay, anabolism is to build up. Catabolism is to break down. And the nature of physics is that when you build something, you have to put energy into it, and when something breaks down, it releases energy. It would take you energy to build a brick building. And then if those bricks fall, they would release kinetic energy. That's just how this works, all right? And the same sort of conversation applies down here. Anabolic reactions are synthesis reactions. They are making something, synthesis to make. And catabolic reactions are decomposition reactions. They break things down. Okay. And in the realm of this conversation, we need to have a conversation on polymers and monomers. Alright, a monomer is a building block. If you have a building made out of bricks, the monomers are the bricks. The polymer would be the building. Okay? A polymer is a big long string of monomeric subunits. For example, starch, which is found in potatoes, for example. Okay? Starch is a polymer of many glucose monomers. Glucose would be monomers. 3,000 glucose monomers make a single starch polymer molecule. Okay? So polymers are large, monomers are their building blocks. So if you take a couple of monomers and stick those together, what you're doing is you're making part of a polymer. And here we have two very important terms. First is dehydration synthesis. The second is hydrolysis. Dehydration, synthesis, and hydrolysis. Now, dehydration synthesis is how your metabolism makes everything it makes. Okay? When you've got nutrients in your bloodstream, you use some of those to fix a cell or to do some sort of bodily maintenance, that is dehydration synthesis. It's called dehydration synthesis because it's dehydration synthesis. Synthesis is to make, so you take two monomers and you make a polymer, that's synthesis. But this is dehydration synthesis. And it's dehydration synthesis because water is pulled out of the system. When you make this polymer, water leaves. Okay, the water leaves. And then, of course, there's hydrolysis. All of your digestive reactions are hydrolysis reactions. What's happening here is very straightforward. You take a polymer and you break it down into its monomeric subunits by adding in water to the system. When you add in that water, it breaks down your polymer into its monomeric subunits. We do this so that we can digest food. Okay, so we can break things down. You can't absorb large molecules like this into your intestinal tract, so you have to break them down into smaller units before you can actually get them into your bloodstream. So dehydration synthesis to make by removing water. Hydrolysis to break by adding water. Okay. Alright. Okay, first and foremost, the carbohydrates. Now, in the realm of carbohydrates, we have the monosaccharides. Okay, of which there are several. We will talk about a few here. So the three major monosaccharides that are worthy of our consideration are glucose, fructose, and galactose. And then, of course, you can't leave out deoxyribose and ribose. Now, again, these are the sugars that we think of. Carbohydrates are the sugars, and these simple sugars are things like glucose, fructose, and galactose. Glucose, you should be particularly aware of because this is your blood sugar. Okay, when somebody says your blood sugar level's high or low, they're talking about glucose. Deoxyribose and ribose you should be very familiar with because these are involved in nucleic acids. Deoxyribose in your DNA and ribose in your RNA. Now, if you take these monosaccharides and start sticking them together, you can make some what are called disaccharides. Okay, these are two parts, disaccharides. You can take glucose and fructose and stick those together via dehydration synthesis, and you can make sucrose. Sucrose is good old-fashioned white table sugar you put in your coffee. If you take a couple of glucose molecules and bind those together, you make maltose. Maltose is very important in the brewing industry for brewing alcohol and spirits. And then, of course, if you take glucose and galactose and bind those together, you can make lactose. And lactose is the sugar found in the milk we feed our young as mammals. So all of these are very, very important for us. And they are all made by taking monomers and putting them through dehydration synthesis to make a theoretical polymer. Okay? And then these are too big for your intestinal tract to absorb, so you have to break those down via hydrolysis to their monomeric subunits. The polysaccharides. So there are a lot of polysaccharides in the world around us. Amongst the carbohydrates that we are capable of making, however, there is only glycogen. Again, there's others that are found in other systems, but glycogen is the only one we are capable of making. This is our glucose energy storage molecule. Okay, when we want to store glucose, when we take in a lot of glucose into our systems, we will store that in our liver. This is typically made in the liver, and that's in the form of glycogen. Yeah, it's just a good long-term storage. The idea here is that for you to prevent glucose from being too mobile in the system, what we'll do is we eat food. That food contains glucose. That gets into our system, becomes blood sugar. Okay, that blood sugar, if you need to have less of it, you'll release insulin and drop your blood sugar levels. Part of the way we do this is by pulling glucose into the liver and making glycogen, which is a storage form. Now, if you have lots and lots of sugar in your system and you've got plenty of glycogen storage, what you will eventually do is you'll start taking that sugar and start manufacturing fat from it. Okay, a high sugar diet will certainly result in a lot of fat being produced. So, sure, that's perfect. That'll do. So insulin drops blood sugar levels. That's a fact. Alright, let's go here. The lipids. So the classic lipid for our purposes is the triglyceride. It is called a triglyceride for a reason. It's a glycerol molecule with 1, 2, 3 fatty acid chains attached to it via dehydration synthesis. You make triglycerides via dehydration synthesis, and the triglyceride is the basic lipid that we will be discussing. Now, in the realm of triglycerides, there are two forms, what are called saturated fats and unsaturated fats. This is very simple to understand. Saturated fat will always be, well, let me say this. A saturated fat is typically of animal origin. It tends to be solid at room temperature. Unsaturated fats tend to be of plant origin, and they tend to be oils. Okay, they tend to be liquids at room temperature. And the reason for this is pretty straightforward. A saturated fat will be solid at room temperature because the tails, all right, these fatty acid tails, are completely saturated in hydrogen. They're just packed with hydrogen. And because they're packed with hydrogen, the tail will be straight. And I don't know if you've ever played Tetris, but the straight pieces are a lot easier to line up than are the crooked ones. So at room temperature, these stack up like blocks, and they make a solid. All right? They make a solid. Whereas an unsaturated fat, being if it has one kink in the tail or lots of kinks in its tail, an unsaturated fat, because of the kink in the tail here, this will be liquid at room temperature. It just doesn't want to stack up at room temperature like a saturated fat would. So it will remain liquid as a result of a double bond and a lack of hydrogens as a result. Now, worthy of our consideration are also trans fats and omega-3 fatty acids. Trans fats are considered very, very bad for us. omega-3s are considered heart healthy. The idea is that if you're consuming 1 to 2 meals that are fish-based a week, which is where we get a lot of omega-3s, you can get omega-3s through all sorts of sources these days. So you could buy fish oil pills. You can get bread that's enriched with omega-3s. Heck, the milk I give my kids is enriched with omega-3s. But omega-3 fatty acids are known to be heart healthy. They decrease your risk of heart disease, whereas trans fats, which are in this realm, are very much considered to cause heart disease. And I want to mention why that is just a little bit. So what a trans fat is when you take a cis fatty acid and you partially hydrogenate it and turn it into a trans fatty acid. Basically what we're doing is we're taking the kink tail of a lipid, a liquid lipid, a oil, taking that kink tail and artificially straightening it a little bit. Now it's not fully straight, but it's straight-ish, let's say. And because of this, there's actually nothing in nature that looks anything like it. And because there's nothing in nature that looks anything like this, there are no enzymes that are capable of breaking it down. You can make a food using trans fats, and that food is going to remain for the foreseeable future. Okay, it's not going anywhere. They don't break down. Trans fats don't break down. There are bacteria struggle with trans fats. You know, your intestinal tract, when you absorb trans fats, they just kind of stay in your system for a prolonged period. And what eventually happens is that, well, frankly, they can lodge into the walls of your arteries and lead to atherosclerosis. Trans fats are, according to the CDC, I think I've got a quote here, FDA. The FDA says that trans fats are, quote, not generally recognized as safe. So the take-home message here, I suppose, is to stay the hell away from trans fats. All right? These are very dangerous. They will lead to heart disease. So don't eat food that says partially hydrogenated or trans fat or what have you. Okay, just try to steer clear. It is a bad idea. All right, phospholipids. So a phospholipid is a phospholipid for a reason. What we have here is phosphorus heads stuck to polar tails, or non-polar tails, I should say. So this looks a lot like a triglyceride, glycerol backbone 1,2 fatty acid tails, just like a triglyceride, but instead of having a third fatty acid tail, a phospholipid has a phosphorus group sticking off the other end. And this is a weird molecule. And it's a weird molecule for a reason. What we have is this polar end stuck to a non-polar end. I don't know if you remember us talking about fats and oils, but fats tend to be non-polar, covalently bonded, non-polar, so they don't bond to water. But a phosphate group, which is polar, gets stuck onto the end of this, and that means that this molecule is capable of binding to both water and oils. Water and oils, which is interesting to say the least, all right? Because of this, this membrane tends to form our cellular membranes. Or did I say that correctly? This particle tends to form our cellular membranes. We have these big old carbon chains with hydrogen, and what you'll find is that they have hydrophobic heads and my dyslexia is going crazy today. What we have are these hydrophilic heads and hydrophobic tails, okay? So they will form up in what's called a double membrane, a phospholipid bilayer, and this will make your cell membranes. Hydrophilic heads sticking off the outside and in the inside, and then the core of this is going to be these hydrophobic tails. Now, what does this mean? It means that if I am a polar molecule, I'm going to struggle to get through the core of this. If I'm a non-polar molecule, I'm going to struggle to get through the heads. Okay? Basically making your cell membranes selectively permeable, which is the key term here. These phospholipids make your cell membrane selectively permeable. And you can see this happen in real time. These are really good at forming membranes. If you go and buy bubbles, you know, like kids blow bubbles, if you go and buy the fluid for bubbles, what that is just, in essence, molecules that mimic phospholipids. Dawn dish soap, these are molecules that mimic phospholipids. And the reason is very simple. You have a greasy pan, you can't get the grease out, so you pour a little bit of Dawn dish soap in there and stir it up, and the grease comes right on out. Because If you're just pouring water over oil, they can't physically bind to one another. But if you mix in some phospholipids, the hydrophobic regions will bind to the oils, and the hydrophilic regions will bind to the water and allow you to wash oils off of pans. Okay, soaps are based off of this type of molecule. Cholesterol. So if I were just to ask you if cholesterol is good or bad, you'd probably say cholesterol's bad, but that does not even remotely tell the whole story. Cholesterol is very, very important for you, and you have to have it. In fact, if you don't have enough in your system, your body will manufacture cholesterol from scratch. All right, you have to have it. It's very, very important. Cholesterol is, are we listening? The precursor molecule to all steroid hormones. Things like testosterone and estrogen. Okay? You must have cholesterol to then make other steroid hormones that, for instance, give us our secondary sexual characteristics. And there's other steroid hormones that are worthy of our consideration. Things like vitamin D as an example. So vitamin D, we must have this to survive. Vitamin D allows us to absorb or assists us with the absorption of calcium from our intestinal tract. And this is, in fact, a steroid hormone. Cholesterol has other roles. Do I have an image of this? No, I don't. Cholesterol is found in our cell membranes. And basically, if the cell membrane gets too cold, it can become rigid, and cholesterol prevents that rigidity from taking place. And also, if our cell membranes become too hot, they can become too fluid and rupture, and cholesterol actually dampers the effect of heat on cell membranes. So cholesterol is a major stabilizing factor when it comes to your cellular membranes. I don't really think we necessarily have to talk about steroid tests in athletes. You know that there's a lot of steroid use in the athletic world, and it gives rise to some really crazy effects, but I am not going to go through those. Let's jump straight into proteins at this stage. Alright, proteins are very diverse. So again, I probably said this before to you, but when I see you, everything I see is protein. Your hair, your skin, your nails, all of it. Your muscles, all of it. It's all protein. You got protein all over the place. Your enzymes, like your digestive tract, contractility for your musculature, oxygen carriers like hemoglobin, it is all protein. And protein is made out of minor monomeric subunits called the amino acids. Okay, amino acids are the building blocks of proteins. You string together 500 or 1,000 amino acids, you've got part of a protein on your hands. Okay, they are made of amino acids. Made through dehydration synthesis. Now, here's an important point. The term here is a peptide bond. Now, we just add this term in. This is just how we do things when we're dealing with protein. When you say that you bind one amino acid to an adjacent amino acid, that's a covalent bond. Okay, we're making a covalent bond here, but because this is making protein, we call it a peptide bond. A peptide bond is what we call covalent bonding between amino acids. Okay, when you're making protein, we call them peptide bonds. There can be dipeptides or tripeptides or polypeptides or what have you. Okay, so there's a little bit of variety here. Now, proteins also come in a variety of structures. There are primary structures, secondary, tertiary, and quaternary, and they go something like this. All right, a primary structure is basically just a string of amino acids. This is going to be made in your rough endoplasmic reticulum of your cells. You'll have molecules come together, they just start stringing amino acids together, one after the next, after the next, after the next, amino acid, amino acid, amino acid, amino acid, amino acid. And what they're doing is they're making a primary string, a primary strand. This primary strand can align itself sort of, up and down like this and make a beta pleated sheet. Okay. This primary strand can wrap itself up in coil to form a secondary structure called an alpha helix. And then those can secondarily coil and modify their shapes to become a tertiary structure. And then you can bring together several of these tertiary structures and make a quaternary structure if necessary. This can happen. Now, all of this is peptide bonds. They are covalent. All the rest of this, these unique shapes that are taken on, those are all done by hydrogen bonds. All right? They are all hydrogen bonded. Hydrogen bonds cause these weak links between parts of a beta pleated sheet or an alpha helix. Hydrogen bonds cause these tertiary structures to stick together in their unique shapes. Hydrogen bonds are very, very important for us. And this leads to one more term being denatured. Okay, if you grab a hot pan and you burn yourself, you end up with a stripe across your hand or some blisters. What you've done is you have denatured the proteins in your skin. You've busted them up using heat. If you go and you eat a salad, you put nothing but vinegar on that thing and it eats up the inside of your mouth, all right, your oral lining. What you've done is you've used acidity to denature the proteins in your skin surface. This concept, what denaturing is just busting up the hydrogen bonds. The covalent bonds tend to stay in place because they're very, very strong, but these hydrogen bonds are weak. So you can apply a little bit of heat or a little bit of acidity to an enzyme, and it completely screws with its capacity for function because it denatures it. Okay, it causes it to become mildly denatured. Yep. All right. Enzymes, a substrate. Yeah, we need to talk about it. So what the heck is an enzyme? Enzymes are biological catalysts, okay, biological catalysts, and what they do is typically they just what's called lower the activation energy for a chemical reaction to take place. Enzymes increase the likelihood of a chemical reaction. Yeah, we're doing good. Enzymes increase the likelihood of a chemical reaction. What they do is they act upon a substrate. This could be a molecule or a group of molecules, and it will either break that down or build that up. Now, an important point to be made is that all enzymes have a naming constraint. They all end in ASE. For instance, acetylcholinesterase is an enzyme that breaks down acetylcholine. Okay, and that's really good enough for me. Like the activation energy is all well and good. So the goal here is for you to understand that enzymes make chemical reactions take place more easily. I want you to understand that enzymes are very, very accurate. They only work on certain molecules, so they have what's referred to as an active site, and it's lock and key. It's incredibly specific. In fact, if I'm an enzyme that breaks down lipids, I cannot have any capacity to augment proteins. Okay, if I'm an enzyme that breaks down starch, I have no capacity to break down any other carbohydrate. Okay, so they are very, very specific. Highly specific lock and key, and enzymes are not consumed by the reaction. The idea is that an enzyme can catalyze a reaction after reaction after reaction after reaction after reaction. And then last but not least, we have the nucleic acids. So nucleic acids are made of, well, let me say this. In the realm of nucleic acids, what we're talking about here is like your DNA, okay, deoxyribose nucleic acid. And DNA is made of certain building blocks, or I should say all nucleic acids are built of certain building blocks: adenine, guanine, cytosine, thymine, and uracil. The big ones, the large ones, 2 rings, are called adenine and guanine. These are the purines. And the small ones, which are single ring structures, like thymine here, are cytosine, thymine, and uracil. Now, let's not get too deep before we get to the next slide. Is this, yeah, it's good. All right, so 2 nucleic acid classes, DNA and RNA. This is a quick comparison. We're going to go into way more detail on this a little bit later on. But for our purposes, DNA is always found inside the nucleus of your cells, whereas RNA is made in the nucleus but does its job out in the cytoplasm, typically in the rough endoplasmic reticulum. DNA is certainly replicated for cell division. RNA is going to be made when you need proteins. RNA is a code for making proteins, okay? Well, let me rephrase that. I'm just going to lay this on you. We're going to talk about this more in the future, but I feel like a little foreshadowing could help. DNA is like a cookbook for how to make protein. DNA is not fancy. It doesn't do anything terribly amazing. There's no mystery involved in anything DNA related. DNA is simply a code, a cookbook for how to manufacture proteins needed by your body. That's it, period, the end. How do you make those proteins? You have to get a copy of your genes, a small subunit of your DNA, out into the rough AR, rough endoplasmic reticulum, for the manufacture of said protein. How do you do that? You make a little copy of a piece of your DNA, and those copies are called RNA molecules. So RNA molecules are little pieces copied from DNA. Now, DNA contains adenine, guanine, cytosine, and thymine. RNA contains adenine, guanine, cytosine, and uracil. In RNA, we replace thymine with uracil, pretty straightforward. And another major difference between DNA and RNA is that DNA contains deoxyribose as its sugar. Okay, you can see that here, these are the sugars we're talking about, contains deoxyribose, whereas RNA just contains ribose. Now, I will also point out ATP real quick. It's not necessarily a nucleotide, but it looks so much like one that we're going to consider it. Okay, what we have is in ATP, this is our energy molecule. So if you're listening to me right now, your ears are capable of firing off messages to your brain because they contain ATP. All right, you are capable of standing up and walking around because of ATP. Like ATP is your energetic molecule. So what the heck is ATP? Well, it's adenosine triphosphate. What we have here is adenine and ribose, and 1, 2, 3 phosphate groups at ATP, triphosphate, triphosphate. 1, 2, 3, triphosphate. And if you look at this, it looks a lot like this. Adenine, let's do it like this. Nucleotide, sugar, or nitrogenous base, sugar, phosphate. Nitrogenous base, sugar, phosphate. The difference is the two others that are bound here. Now, the energy in ATP is stored in the final phosphate bond. So what will happen is when you use ATP, it kicks off the phosphate, which is kinetic energy, and that will give you ADP, adenosine diphosphate. That ADP can be re-energized into adenosine triphosphate, and then the body can use it again and again and again and again and again. This is done, again, in your system, in your mitochondria, through aerobic cellular respiration most of the time. And that's that, folks. So let's stop it there, and you can expect the quiz on this. Actually, I think the quiz on this is already up. You can expect the next lecture pretty soon. All right, thanks, guys.